This is an overview of an article entitled Large-Scale Phenome-Wide Association Study of PCSK9 Variants Demonstrates Protection Against Ischemic Stroke, published in the July 2018 issue of Circulation, Genomic and Precision Medicine. PCSK9 inhibition is a potent new therapy for hypercholesterolemia and cardiovascular disease. Although short-term clinical trial results have not demonstrated major adverse effects, long-term data will not be available for some time. Genetic studies in large biobanks offer a unique opportunity to predict drug effects and provide context for the evaluation of future clinical trial outcomes. In the paper under discussion, the authors performed a variety of association analyses with PCSK9 variants, principally the missense variant RS11591147, in up to 337,536 individuals of European ancestry in the UK Biobank cohort. First, they started with 11 predefined, hypothesis-driven cardiovascular phenotypes. Second, they performed an unbiased Phenome-Wide Association Study, or PheWAS, with 278 manually curated phenotypes with the intent of assessing for previously unappreciated effects of PCSK9 inhibition. Finally, they utilized a relatively new approach called TreeWAS, using a comprehensive tree-like phenome map of ICD-10 codes in the electronic health record. They performed TreeWAS for the single variant RS11591147, as well as a score comprising 6 different PCSK9 variants. More details on the TreeWAS methodology are available in the published article. Here are the association results for the predefined hypothesis-driven cardiovascular phenotypes. Of note, the authors randomly assigned UK Biobank participants to discovery and replication groups, 225,000 and 112,000 in size, respectively. This allowed the analyses to take place in 2 stages, even if there was not a true independent replication performed. In the primary analysis, there were significant associations with use of lipid-lowering medications, coronary heart disease, and ischemic stroke. Of note, there was no association with type 2 diabetes in the primary analysis, but a secondary analysis in which the authors adjusted for lipid-lowering medication use found a significant association with diabetes. Further, this association was only observed in those individuals using lipid-lowering medications. Here are the significant associations that emerged from the unbiased, hypothesis-generating PheWAS analysis. Notably, all of them related to cardiovascular or metabolic diagnoses. Here are the results of the TreeWAS analysis for the single variant RS11591147, although it should be noted that TreeWAS results for the 6-variant score were highly similar. All of the phenotypes above the gray line can be regarded as statistically significant. Again, the significant as well as the suggestive signals were all related to cardiovascular and metabolic diagnoses. In conclusion, the authors obtained strong genetic evidence that reduced PCSK9 activity is protective not only against coronary heart disease, but also against ischemic stroke, defined here using infarction and occlusion of relevant blood vessels. They did not find evidence of cognitive side effects of PCSK9 variants, which has been a concern for PCSK9 inhibiting therapies, but has not been observed in clinical trials to date. Nor did they observe associations with other concerning outcomes such as cataracts, heart failure, and atrial fibrillation. Finally, the authors observed an association with type 2 diabetes in a stratified analysis of individuals on lipid-lowering medication, consistent with prior genetic analyses, although clinical trials have not uncovered any effect on the incidence of diabetes so far. Thus, This work predicts that PCSK9 inhibitors will be effective in reducing ischemic stroke in the long term while being largely free of non-metabolic effects.